As a quick review, to this point we've introduced how to analyze various types of data. We started with one continuous variable, and we learned how to run t-tests and z-tests and generate 95% bootstrapped confidence intervals. We also know what type of displays to use with them. So if we have a continuous variable, one of our displays, our preferred displays, is a histogram. We then move to working with two variables. So we have one categorical variable, that's our grouping variable, and it's the independent variable. And then the second variable is a continuous or measurement variable. That is our dependent variable. And with those types of data, we preferred to display data using a box plot or a box and whiskers plot, or perhaps a plot of means with confidence intervals. And we analyzed our data several ways depending on our questions. So if we were interested in comparing means, we could use an ANOVA or perhaps a t-test if our groups was only two groups. If we were interested in comparing medians, we could run a Kruskal-Wallis test. And we also learned how to check assumptions of ANOVA, testing for equal variance, and so forth. What we're going to do now is go back to working with one variable, but this time instead of that variable being continuous or a measurement variable, we're now going to look at the case where our variable is a categorical variable. So when we work with one categorical variable, we have two types of displays that we can choose. One type is a frequency table or an enumeration table. This is going to list our count or our frequencies, and we did this already in R. We use the table function, and remember with the table function we pass it a vector. So if that vector or that variable is in a data frame, we would have to use our data $variable format to generate our frequency table. We could also display our counts using bar graphs. Now with these bar graphs, the information is displayed or relayed to the reader as the height and the area of the bars. We separate the bars, we have spaces between the bars to emphasize the categorical nature of our data. And when we do these bars, we don't really have any sort of standard error or variance measure assigned to it because it's just representing a frequency or a count. However, we could generate some bootstrap confidence intervals to add a measure of dispersion to our graph. Excel makes really nice bar plots, so if you wanted to do a bar plot for a paper, I would suggest generating our table in R and then transferring that information to Excel to make your box plots. But if you're just doing this to visualize the data prior to running any sort of statistical test, then we can make it in R using the bar plot function. And for the bar plot function, we have to pass it a table, a frequency table. Once we make our figure, we may be interested in asking if our counts equal some expected frequencies. These in general, these types of questions rely on goodness of fit tests to determine significance. All of the goodness of fit tests that we learn about are comparing observed frequencies to some expected frequency. So all of them have the same general null and alternate hypothesis. The null hypothesis is that our observed frequencies match what are expected, and our alternate is that the observed frequencies do not match what is expected. Now, much like in ANOVA, it doesn't tell us which frequencies differ, it just says at least one or probably two frequencies, observed frequencies, do not match the expected frequencies. We can get a little bit more specific with our hypotheses depending on the biological question of interest. So our biological question of interest may be related to some theory or some idea. So I'll use Mendelian genetics. Perhaps we did a dihybrid cross. So we took two purebred strains, generated the F1 generation, and then crossed the F1 generation to each other, and we observed a frequency. We observed 4 phenotypes. And our question is, Did our observed frequencies match what we expect? Well, what do we expect? It comes down to what we think the genes are. So if we think they're autosomal in nature, we would expect a 9 to 3 to 3 to 1 ratio. So we would compare our observed frequencies to the expected 9 to 3 to 3 to 1 ratio. That form of a hypothesis is an extrinsic hypothesis. We derived our expected frequencies based on some underlying theory or some hypothesis that's external to the data. We didn't rely on the data itself to generate the expected frequencies. We derived the expected frequencies based on our own understanding of how things should occur. Now, with the extrinsic hypothesis that I just gave, we could change our hypothesis not just to say observed frequencies match what we expect, but we can be more specific and say the observed frequencies match a 3 to 3 to 1 ratio, or 9 to 3 to 3 to 1 ratio, and so forth. The other type of hypothesis is an intrinsic hypothesis. An intrinsic hypothesis generates expected frequencies just like the extrinsic hypotheses, but with the intrinsic hypotheses, we had to calculate something from the data first before we can generate our expected frequencies. So the type of questions that this would ask is usually, are the data random? In which case, we utilize our data to get some of the summary statistics that are then used to generate our expected frequencies. When we use intrinsic hypotheses, for every statistic that we estimate, we lose a degree of freedom. And this will become important when we come back to fitting distributions. So, of our goodness of fit tests, regardless of the hypothesis, we've got three main tests that we're going to be introduced to. We have a binomial test, we have a chi-square test, and we have a g-test. The binomial test is only going to be used when we have dichotomous data. So, yes or no outcomes, infected, not infected outcomes, male, female, and so forth. We learned about binomial distributions earlier in the class. Feel free to go back and look at how we calculated probabilities. Second broad types are the chi-square test and the g-test. These can be used when we have two outcomes or more outcomes. Also, we have randomization versions of each of these chi-square and g-tests. So here's a working example to demonstrate all three of these tests. The data file is posted as flies icolor.csv. And what I'm going to ask you to do is pause the video, download this file, and read it into R as flies. So pause the video, import it into R, and then come back. So this data set represents counts of fruit fly eye colors. So here's our problem. Gina Leal claims to have found a new mutation that affects eye color in flies. She postulates that this is an autosomal mutation. To test this hypothesis, she crossed a purebred wild type with a purebred mutant. She collected the F1 generation and allowed them to mate. The F2 generation exhibited 315 red-eye and 95 white-eyed flies. So the question, is this an autosomal mutation? So that's our biological question. Is this an autosomal mutation? What we have to do is generate the hypothesis. So our null and alternate hypothesis is simply, did we observe what we expect? That's a simple way, but to get a little bit more specific, the question is, well, what do we expect? Well, it goes back to the problem. If we expect if we know this is an autosomal mutation, or we suspect this is an autosomal mutation, then we should expect a 3 to 1 ratio, 3 red-eyed fly to 1 white-eyed fly ratio. So what we can do is compare the ratio. Did we get, and say our null hypothesis is 3 red to 1 white. Alternatively, we can convert this to a percentage to say, is the probability of being red equal to 0.75, or is the probability of being white equal to 0.25? So in both cases, they mean the same thing. Here's our null hypothesis. We use this null hypothesis to generate our expected values, and then we compare our observed to the expected values. Our alternate then is that it's not equal. because we didn't specify any sort of direction. So our options are all goodness of fit tests, and we can run a binomial test, chi-square test, or a g-test, or randomization versions of the chi-square and the g-test. So let's start with the binomial test. Binomial test is a goodness of fit test for two outcomes. It is essentially asking how likely is it that we would observe our data if the probability of success is actually equal to p. This p is usually going to be derived from externally to the data. So we would have some underlying reason to suspect our probability of success is this proportion. This test is an exact test. It's an exact test because we use the binomial distribution to actually calculate the probability of getting data as extreme or more extreme than what we actually observed. So we kind of did this when we did the coin tosses. So heads or tails. And we said we flipped the coin 10 times. We had 8 heads. Is this coin fair? Well, our p-value is going to be the sum of the probabilities of flipping 8 heads, 9 heads, or 10 heads, and then multiplying by 2 to get the two-tailed version. So we actually calculate the exact probabilities. This test is going to be preferred when we're working with two outcomes because it is an exact probability test. Now, what are the assumptions? Well, the assumption is that we're working with the dichotomous variable and that our outcomes are going to be mutually exclusive of each other. So if we have 2 coin tosses, the probability of flipping a head on both is going to be independent of what we flipped previously. And when we flip a coin, we can't get both heads and tails. I put this as an assumption. It's a requirement. We can't do this test if we don't have a dichotomous variable. The important assumption is that we're dealing with a random and independent sample. So how do we do a binomial test? Well, we could do it the long way by using p-binom and calculating all the probabilities, or we can use the function binom.test. So binom.test is going to take the number of successes that we have, it's going to take our total sample size, and it's going to take our probability of success. And then we could do an alternative. So if we have if we suspect that this is not an autosomal mutation and that perhaps we have sex linkage going on, then perhaps we think that the probability of success is going to be less than 0.75 for the red-eyed flies. Well, we can do the direction, but normally our alternative is going to be two-sided. So how would we do it for the fruit flies? Well, we have to first look at our fruit fly table. So in R, we can view the frequency table by using the table function. And what we see is that we get 95 white-eyed and 315 wild-type flies, or red-eyed flies. So what I'm going to test is the probability that getting a red-eyed fly is equal to 0.75. So I would do binom.test, 315, that's what we observed. Our out of number is the sum of these two, so 95 plus 315. And our hypothesized probability of success is 0.75. We don't set direction, so alternative is equal to two-sided, which is the default. And then we get this output. So the output tells us everything that we need. It tells us the name of the test. We ran the binomial test. It doesn't give us a test statistic, and it doesn't give us a degrees of freedom because we calculated an exact probability. So we have our test name, and then the third thing we're going to have is this p-value. So the p-value test is our data likely given that the probability of success equal to 0.75. Well, p-value is greater than 0.05, so we failed to reject our null hypothesis. Our null hypothesis is that our success probability of red eye is equal to 0.75, so we conclude that yes, the probability is equal to 75%. Our write-up then. This is how I would use it for the binomial test. A total of 410 fruit flies were counted, of which 76.8% exhibited wild-type eyes. The proportion wild-type is approximately equal to 75%. Binomial test, p is equal to 0.42. does it matter if we use wild type or white eye to get our p-value? No, it doesn't. It's because our binomial is symmetric around the probability of success. And since we have two outcomes, if we're testing against 0.75, well, that's the same thing as testing against 0.25. So here's my RMD file that I've created for this presentation. I have a library section for the libraries. We'll need that a little bit later. I have my flies imported. I've got a function spot, which we'll need that later, and then I have my binomial test that we just did. So our binomial test is used when testing dichotomous variables. It tests if the probability of success is equal to some value. So with our table, with our fruit fly table, this is my wide-eyed and wild type frequency table. We know that we observed what we actually expected for an autosomal mutation, but what if we did our wide-eyed? Well, again, it's binom.test. We list our successes, which is 95. We list our out of number, which is the sum of those two outcomes, and our probability of success is 0.25. When I run this, you will see that I get the exact same p-value, so our conclusion is the same. So let's do one more test. Let's do a practice problem. Let's say we observed infected fish. So fish in a pond exhibit a infection rate of 0.15 or 15%. All right, and we went out to this pond with an idea that perhaps it's going to be equal to 10%. So our question, is this infection rate different than 10%? So pause the video and try to do a binomial test with this. Now, if you struggled, no big surprise, because what we have to do is come up with our observed and out of number. So, well, let's revise this. Let's say 15%. So we have 15 infected out of 100 fish. And now we can run our binom.test. We are going to do 15. The out of number n is 100 fish total. Our probability of success is equal to 0.1. That's what we are testing. And we're going to do two-sided test. So in this case, our p-value is 0.09. We would conclude that the infection rate of infection is not different from 10%. And we have a binomial test, and we report our p-value. We can test these data using chi-square test and a g-test as well, even though binomial test would be preferred. So chi-square test you have been exposed to in previous classes. Chi-square test is normally calculated by hand. It's easy to calculate by hand, and that's why we often introduce it in the freshman classes. So just as a quick review, again, chi-square test compares observed to expected frequency data. The expected frequencies are usually generated by the researcher. Our test statistic is a chi-square value, and we calculate it by taking the sum of the observed minus expected squared over the expected. In Excel, we normally set up a column where we have our column of observed, column of expected, and then we go stepwise to get this a column of these values, and then we add it up to get our chi-square. We're not going to do it in Excel. I'm going to show you how to do it in R. Now, the assumption of the chi-square is that we're working with a random independent sample. No big surprise, it's basically the same for all of our tests. But that we have these other two situations. None of the categories can have an expected frequency less than 1. When they do, it's going to really throw off our chi-square test statistic. Also, no more than 20% of our categories can have an expected frequency of less than 5. So low expected counts in our cells, in our categories, is problematic for this chi-square. The problem comes because it's going to increase our chi-square, and that means then our chi-square is not going to be following the actual chi-square distribution. Our degrees of freedom for this test is the number of categories minus 1. And we'll come back to this because if we're doing a goodness of fit test where we estimate something from our data, we're going to actually have 1 less degree of freedom, or perhaps 2 less. So how do we do a chi-square test? In R, there are 3 different ways to do a chi-square test, and they all can generate different results. So that we might not actually be doing the correct chi-square test that we're trying to do. The way that we're going to run it is this: chi-square.test x, p, correct equals false. All right. So what it's going to do is take a vector of observations, x, and it's going to compare it to the vector of expected probabilities. So we give it this probabilities, and then it's going to run a chi-square test. This correct equals false refers to Yates continuity correction. This is a correction that tries to get our chi-square closer to the actual chi-square distribution. We would use this if we're concerned that we are deviating a little bit. If I was only doing one chi-square, I would probably use Yates continuity correction. But if I was going to do a sequence of chi-square tests or a sequence of g-tests, I would not use any continuity correction. So if we set correct equals false, then our chi-square value and our chi-square test is going to produce the results exactly the same as if we did it by hand. If we did correct equals t, then our chi-square and our p-value is going to be different. So my suggestion for this class is just set it to false. So if you wanted to go back and check it by hand, you could, and you'll get the exact same values. Now what about these other two? These other two chi-square tests we give the data in a slightly different way. So if we give it a matrix of observed and expected, then it'll run a chi-square test, but it'll actually run a contingency analysis. So it'll compare the pattern of observed and expecteds, and we'll get to the contingency test. Same thing with this way. So instead of passing in a matrix of observations, we actually give it a vector of observed values and another vector of observed vectors. We'll do these contingency analyses in a little bit. So let's go back to our chi-square test for the fruit flies. Now when we do this chi-square test, we're going to give it a vector of observed values and then a vector of probabilities. It is very important that the vector of probabilities that we give this function occurs in the same order as our vector of observations. So with this vector of observations, I gave it the wide-eyed flies first, and then the red-eyed flies. So our probabilities then have to be the probability of our wide-eyed flies and then the probability of our red-eyed flies. When I run this, I get this output. So the output says we ran a chi-square test. It generates our test statistic, our chi-square test statistic. It tells us how many degrees of freedom, which is the number of categories minus 1, and it generates our p-value. So just like any other chi-square test that you may have run, we look at the p-value. It's greater than 0.05, so we observed exactly what we predicted. A write-up can be a little bit more extensive based on our experiment. So here's an example write-up where I kind of describe what we did. I reference the table of frequencies, and then I get to the actual chi-square test. So the analysis suggests that the mutation is autosomal in nature. Our observed matches what we expect. The expected was derived based on the assumption that it is autosomal. So therefore, since our observed matches the expected, then our observed does represent an autosomal gene. Chi-square, test statistic, our degrees of freedom, and our p-value. So here's my R. We're running a chi-square test. This is going to test if our observed matches expected counts. So we're going to have to give it both our observed and expected. I also say watch out for small expected values. That's part of our assumption for the chi-square tests. So just like before, we did table. We see 95, 315. All right. And for the chi-square test, instead of making a vector, you can actually pass the table. So I know that this table function returns white first and then wild type. So I'll set up my probabilities in that order, and when I run it, I get the exact same output. So for practice, let's say we performed a dihybrid cross to determine an inheritance pattern of eye color. You observed 318 red, 107 orange, 114 brown, and 48 wide-eyed flies. Are these two genes for eye color autosomal in nature? So, just as a quick review, if these are autosomal, then we would expect a 9 to 3 to 3 to 1 ratio. 9/16 should be red eye, 3/16 should be orange, 3/16 should be brown, and 1/16 should be white. So, get your chi-square, run it here, and when you're done, come back and let's see if you get the same thing that I do. Alright, so hopefully you had an easy time doing this. It is simply chi-square.test. We pass it our vector of observations, so 318, 107, 114, and 48. We then have to pass it our vector of probabilities. Now, I could have calculated the exact decimals, or we can utilize the calculator function in R and say I observed 9 out of 16 should be red, 3 out of 16 should be orange, 3 out of 16 should be brown, and 1 out of 16 should be white. And I will use correct equals false, so that if we did this in Excel, we would get the exact same chi-square value. So I get this output. So what is our write-up? Well, our write-up goes back to our question. Are these genes autosomal in nature? Yes, the two genes appear to be autosomal in nature. And we ran a chi-square test. Our chi-square value is equal to 4.16. Degrees of freedom is 3, so number of groups minus 1, and our p-value is equal to 0.24. The last goodness of fit test is a g-test. So a g-test is a likelihood ratio test, or a maximum likelihood method. This is a little bit different than a regular chi-square test in that it uses a likelihood ratio. So it calculates a ratio of how likely our observed data is relative to the predicted, or vice versa. We can do it in either direction. So predicted versus observed, or observed over predicted. The difference just comes into whether or not this likelihood is a negative value or a positive value. So likelihood is a measure of how well our actual data matches predicted data. Our predicted data assumes that the null hypothesis is true. So our likelihood then is going to range from 1 all the way up to negative infinity if we have it set up like this. So 1 would represent a perfect fit. Negative infinity means that there is no fit whatsoever. So it gets smaller and smaller and smaller as or larger and larger, larger, again, depending on which one we put it up at the top. So as we deviate from our expected, we deviate from 1. Nice thing about likelihoods is that they're additive, so just like our ANOVAs. One reason why I suggest using ANOVA is that we can partition out the variance. So as we have additional independent variables, we can figure out the contribution for each of those variables, and that contribution was the sum of squares. Well, the likelihoods work in much the same way. So if we do a goodness of fit test, a G test, what we could do is look at the contribution based on the likelihood for each of our variables and come up with a measure of how important each of our observations is. Are we going to do this? Not for this class. We will do it for graduate level class and start to go that next step to say, okay, our observed doesn't match the expected. Let's figure out which ones are different. So the G test uses a G, or a likelihood ratio. That G approximates a chi-square test, and it approximates a chi-square when our sample size is large, meaning all of our expecteds are larger than 10. So what we can do is calculate our G and then compare it to a chi-square distribution to obtain our p-value. Our degrees of freedom is going to be the same. All right, it's number of categories minus 1. But instead of reporting it as a chi-square, we're going to report it as a G. So the test statistic is a likelihood, and here's our likelihood that we would use. So if we were to do it by hand, we would set up our Excel column with this equation, then we'd add up that one column and multiply by 2 to get our G. We may have to use a negative to get a positive G, or we might not. It again depends on if we put the expected over the observed or the observed over the expected. And as I put down here with our likelihood ratio, they're equal. The only difference is the negative. So if we have a 0 as an expected value, then this becomes undefined, but we would be able to calculate it like this. Likewise, if we have a 0 in an observed category, but we should have seen something, then this becomes undefined, and we can calculate G using this way. So the test statistic G is actually pretty good, and it's going to be preferred over a chi-square test because of some of these properties. They can handle smaller expected values better than a chi-square. How do we do it in R? Well, here's the difficulty. There are a couple different packages that include the G test in it. So a VCD package, we're going to use this because the G test is built into a test of independence, so contingency analysis, and we'll do that. And it's also built into this good fit function, which is used when we try to fit our data to a discrete distribution. So if we're fitting our data to a binomial distribution, or if we're fitting our data to a Poisson distribution, we can generate a G. We can perform a G test on it. There's two other packages, Desk Tools and RVA Memoir. These two packages include a G test that we would run, but in order to run those, you have to install these packages. And I found that sometimes these packages, especially this one, could be difficult to install because the packages were made specifically for a book. So that's what this was. It was made to follow a statistics book. And as our versions get later, sometimes these packages are no longer kept up. So our alternative, instead of using G-test that's built in into one of these packages, is that we can write our own script, and that's what I did here. So this is going to be our function for the G-test. It's going to take our vector of counts, so it'll take a table or it'll take an actual vector of counts, and it'll also take our vector of probabilities that we give it, and then it'll perform a G-test and it'll give an output. So what you would do is take that G-test script and copy it and paste it into our function part of that R Markdown document. So here's our R Markdown, and we used this function spot before for a couple other things. And what I could do is I could copy the g-test stuff and put it here. Now, when I run this chunk, our g-test appears. And I've written this g-test to follow the same format as a chi-square. So if you know how to run a chi-square test, then you know how to run a g-test. I will note something of how I did this probability. So this probability will say that if we don't give it a vector of probabilities, then it will test if the probabilities are equal across all of the categories. And the chi-square test does the same thing. So our g-test, I've wrote a script for it. Here it is. You copy it, paste it into your useful function script, and then you have it whenever you want. I had also pasted this into a .r file, which is just a text file, and that's posted on Blackboard. So you can just download that R file, and then you have it everywhere you go. So here's our g-test in action using our fruit fly data. So we've got our table that gives us the order in which we give our probabilities. It's set up the exact same way as that we used our chi-square.test. And the output now tells us that we ran a log likelihood ratio goodness of fit test, or a g-test. It gives us our g, that's our test statistic. It gives us our degrees of freedom, and it gives us our p-value. So much like before, we've got our write-up, and we would conclude that the analysis suggests that the mutation is autosomal in nature. Our p-value is greater than 0.05. There's our g, there's our degrees of freedom, that's our p-value. All right. So here's my g-test. You already saw that I had copied and pasted that g-test function and ran it, so it is accessible. We have our table of fruit flies. We've got 95 white and 315 wild type. That gives us the order in which we would input our p, or the probability, and then when we run our g-test, we get this nice output. So it's not very difficult to run this test. So here's a practice. Community of skunks consists of 60% striped skunks, 20% spotted skunks, 20% hog-nosed skunks. Forgive me if these distributions are all messed up. I made them up. So, regardless, we're going to say it's 60% striped, 20% spotted, 20% hog-nosed. After a year of trapping, you caught 35 striped, 15 spotted, and 10 hog-nosed. Does your catch reflect the distribution of species in the community? So what we are doing is comparing our catch versus distribution, and assuming random catches, then our percentage caught should reflect that population. So go ahead, pause this video, and run a g-test and have a write-up that answers this question. All right, so hopefully you had a very easy time to do this. Test, it's just g.test. That's what we're going to run. We pass it our vector of observed count, so I will use 35, 15, and 10. We also pass it our vector of probabilities, so 0.6, 0.2, and 0.2. And when we run it, we get this output. So what is our write-up? Well, our write-up's based on p-value and our question. It compares our observed to the expected, so our p-value is greater than 0.05, so yes, our observed matches what we expect, which means that yes, our distribution, our catch matches the distribution. So our catch matches the frequency of species in the community. We used a g-test. Our g is equal to 1.08. Degrees of freedom is 2, and our p-value 0.58. All right, so here's one practice problem that you can do on your own. So standard M&Ms come in 6 colors. Well, they have So according to a 2007 web post, color should be distributed with 13% red, 16% green, 14% yellow, 24% blue, 20% orange, and 13% brown. I do say that they said six colors, but I think I've read that M&M's is phasing out the brown M&M's. So here's our distribution. We received several 2-pound 10-ounce bags as gifts and decided to count each color. Does the distribution of colors match what is reported by the Mars company? Well, our data is going to be found in mms.csv, so mms.csv. I want you to download it, import it, view a frequency table, or create a bar graph, and then perform both a chi-square test and a g-test to answer this problem. You should also think, why can't we run a binomial test? So go ahead, download this data file, and run our chi-square and g-test. Just remember, the counts that you pass it have to match the order of the counts, have to match the order of the probabilities that you give it. So hopefully if you had some time and were able to do this. So here's my M&M practice problem. I looked at the table. You'll note that the table listed the colors in order alphabetically. You should have no problem rearranging these into any order that you want using the factor command. You can do that on your own as practice, and if you want, you can email me and I can check your work. It's not super important that you do it for this problem because we can manually specify how the order of these probabilities. So that's our order. We want to run a chi-square test. So chi-square.test. I'm going to pass it this table. So table MMS color, and then I have to pass it the probabilities. So our probabilities have to occur in the order in which they appear in our table. So I start with blue, and then it's brown, and then green, and then orange, and then red, and then yellow. And with the chi-square, I'm going to use correct equals false. I run that, and I get p-value of 0.0235. So what does that tell us? Does our observed match the expected? Well, if our null hypothesis is that the observed equals our expected and our p-value is less than 0.05, we reject that claim. So we conclude that no, our observed does not match what we expected. And we ran a chi-square test. Our chi-square is equal to 12.99. Degrees of freedom is 5. P-value is 0.02. So that's our chi-square test. What about our g-test? Well, g.test. I'm going to highlight this and copy it and paste it. It appears in the exact same order. We get same answer. So I can do my write-up same way, but update that we ran a g-test. Our g is equal to 12.96. Degrees of freedom 5. P-value is 0.02. So we came up with the same conclusion, and that's no big surprise. When we have larger sample sizes for each category, then our chi-square and our g-test are going to be, they will generate approximately the same p-value. There will be some differences because of the difference between a chi-square and a g-test statistic, but by and large we'll get the same conclusions. When we get different conclusions, it is normally due to small sample sizes in one or more categories. So that brings us to which one should we prefer? Well, the chi-square test is traditional, but the g-test is considered better by some authors. So there's two big textbooks in biostatistics that are out there. There's one by Sokol and Rolfe called Biometry. In their books, they suggest that g-tests are better, and they advocate running a g-test. In the other book, by Czar, called Biostatistical Analysis, he says that g-tests and chi-square give the same conclusion, and I agree. They do when we have large expected values, in which case, go ahead and do the chi-square test. It's easier to calculate. You can do it by hand. However, you don't always have the situation when you always have larger sample sizes, or even when we have equitable sample sizes across the category. And for that reason, my recommendation is to go and use the g-test more than the chi-square test. This is especially going to be true if our expected is less than 5 for any group. Just go ahead and run the g-test. You'll get more reliable results. If our expecteds are greater than 5, or I'd say even better, more than 10 across all of our groups, then g-tests and chi-squares will likely give similar results. If we have an observed or an expected value that's less than 3 for any of the group, for any of our categories, then my suggestion is to use randomization methods. And we will get to those. Before we move on, what about our corrections, Yates, or you'll see Williams corrections and so forth? Should we use them? Well, if you're only going to do one test on the data, so if you are only going to test observed versus expected for, let's say, our fruit fly eye color, just do it once and be done with it. Then, yeah, you can run a Yates correction. That's perfectly fine. You'll note that when you run this correction, you're going to increase your type 2 error rate. So you reduce your type 1, but increase your type 2, which is fine. That works. And if I was doing it, yeah, I'd probably use the corrections if I only did one test. But generally, when we run these tests, we're not just running one test. We're going to be interested in trying to identify which of the groups differed. So that means we're going to run some repeated tests, in which case we should avoid the continuity corrections. That maintains the additivity of all of our g-test statistics. So we're going to do a repeated or replicated goodness of fit tests in a graduate level class. This method appears in the biometry book by Sokol and Rolfe. I know the library has this book, so if you're interested in it, you can look it up, or you can just look up repeated or replicated GOF tests, goodness of fit tests, to read about this stuff. So as I said, we do have randomized versions of these tests. These are randomized versions and not permutations because we will generate random data. We're not permuting our data in any way when we have one categorical variable. When do we do that? Well, we're going to use these randomization tests when our traditional tests do not apply. More specifically, when we have small observed or expected counts. And this will occur if we have it in one category or if we have it in a few categories. I also recommend doing these tests when we have very unequal observed or expected across the categories. So in our skunk example, we had 35 and 15 and 10. They're unequal, but they're not drastically different. Let's change it though, and let's say we had 150 striped skunks and then 15 and 10. We've got drastically different counts. This is a case where we should run our randomization tests. So as an intro, just recall that if all of our assumptions are met, then our test statistic, be it a chi-square or a g, should follow a chi-square distribution. As these situations appear in our data, then we will start to deviate from the chi-square. We can fix that by running a randomization test, which basically generates our own null distribution of the test statistics. So it'll generate a null distribution of the chi-square, it'll generate a null distribution of the g itself. The assumption of this test is that our data are random, that they're random and independent. So what we would do is basically take our probabilities, and then R will generate a random sample, which then performs a chi-square on that random sample to get the chi-square test statistic, saves it, and then repeats it over and over and over and over again. We then compare our test statistic to that chi-square. So how do we do it in R? It's actually very, very easy. So in that R script, that chi-square.test function, there is something called simulate.p.value and a b. These two tell R to actually simulate your p-value, meaning perform the randomization tests. So when we set simulate.p.value equals to t, then we can specify how many replicates to run. Normally we run 10,000. When we do this, don't use Yates correction. We want the uncorrected chi-square so that our null distribution is accurate. So how do we do it with our fruit flies? Well, we run chi-square.test, table, flies, $i. We give our p-value. Everything's the same, but this time we do simulate.p.value equals t. Number of replicates b is equal to 10,000. And after a little bit of time, it comes up with this output. You'll note that it still gives a chi-square. That's our observed test statistic. Doesn't give us a degree of freedom because we're not generating our p-value based on some underlying distribution. And it calculates a p-value. The p-value is calculated the exact same way that we did it in our permutation t-test and our permutation ANOVA. It looked to see how many of our random replicates equaled or exceeded this observed chi-square value, and in this case it was about 43% of those values. So based on this chi-square, or this randomized chi-square, we would conclude that the mutation was autosomal in nature, that our observed match what we expect. Number of replicates 10,000, or p-value. I do give this test statistic, and I would give the test statistic even for a randomized ANOVA, but it's not necessarily required. So that's our randomized chi-square test. So let's go back and use a randomized chi-square for one of our practice problems. So here we did our 9 to 3 to 3 to 1 ratio. We did a chi-square test, and that was our write-up. Let's do the randomized version. So I'm going to add a new chunk here. All right, so pause the video, write your script so that instead of doing a classic chi-square test, we do a randomized chi-square test. Pause the video and then come back and we'll check our answers. All right, so hopefully you did it okay. So let me show you how I did it. I'm going to cheat a little bit and copy and paste. And then I'm going to do simulate.p.value equals t and b equals 10,000. When I run that, you can see it runs, it gives this output, and we get the exact same thing. So you will see our observed chi-square, same. Degrees of freedom isn't reported because we generated our own null distribution, and our p-value is pretty close. When our assumptions are met, our randomized p-values will be very, very close to our classical tests. That's the same thing for t-tests, for ANOVAs, and everything else. Also, the larger we are, so the larger the p-value is, the larger the differences we can get, or at least the range of p-values that we can get. The smaller our p-values, the smaller it is, and then it goes back to the coefficient of variation. So you can review what that is and why we would use that, but it applies. It applies. So as you can see, a randomized version of chi-square, very easy to do, and in fact, there's really no reason why you wouldn't use a randomized chi-square whenever you do want to run a chi-square test. So as one more practice, go ahead and run the randomized chi-square on the M&M dataset. And the question, do you reach the same conclusion as a regular chi-square test? And how much did your p-value change? So pause the video, Run your randomized chi-square test on the M&M data set and see if you get the same results. So here's my randomized chi-square test. Same format as a regular chi-square, except I added simulate.p.value equals t, b equals 10,000, and you will note we get the same chi-square value as we should. df is not available, and our p-value is 0.0243, which is very close to this p-value. So do we get the same conclusion? Yep. How much did our p-values change? Not much. No big surprise because we have a small p-value, and because we have basically all of our assumptions are met. What about a randomized g-test? Randomized g-test follows the same principle as the chi-square test. We would generate a random sample based on our probabilities, run a g-test, save the g, and then repeat it over and over and over again so that we get a null distribution of our actual g-test statistic. And then we calculate p-value much the same way, the proportion that equals or exceeds that observed test statistic. R lacks a built-in function for this, but we can write one, and I did. And this is our g.test randomized version. So I called it g.test.rand. It takes the same format as before, so our x vector, our probability vector, and this time instead of b, I used n for the number of replicates. So that way it is equivalent to our randomized chi-square. So it's going to do the same thing as the chi-square test, and it actually generates the data the same way using this R multinomial function. So what you would do is copy that script, that randomized g-test script, copy it and then paste it into your RMD document, right into that functions box, and then you run that chunk so that the g.test.rand appears. And then when we run it, after we run it, this now becomes accessible. So here is where I have it pasted. I've already run the chunk, so g.test.rand is accessible. So let's go down to our g-test. So we did our practice with our skunks. All right. So let's run our randomized g-test version of it. So this one is g.test.rand. It's going to take our vector of observations. So 35, 15, and 10. It's going to take our vector of probabilities. And then it also takes our n, which is the number of iterations. So I will do 10,000. When I run it, I get this nice output that tells us that we ran a randomized g-test with 10,000 iterations, gives us our observed g, which is the same that we calculated before, and it gives us our p-value. And again, our p-values are going to be close because our assumptions are met. So just like the chi-square test, once you have this function, it's actually fairly easy to run the randomized g-test. So go ahead now and run this randomized g-test on the M&M data set and see if you reach the same conclusion as our regular g-test. Pause the video and come back. So I just used the same internals. So table, MMS color, probabilities, I use the same internals as I used for all of my other goodness of fit tests, but this time I used n equals 10,000 and g.test.rand. When I run this, I get this output. G is the same as before, so 12.96, 12.96. There's no degrees of freedom, and our p-value is pretty darn close. Again, If all of our assumptions are met, our p-values should be close, and the smaller our p-values, the tighter our range will be. So let's generate a summary for one categorical variables. When we work with a categorical variable, so a variable that has different frequencies or different groups in it, what we're normally interested in doing is asking or answering the question, does our observed match our expected counts? We would display our data using either a frequency table or a bar plot, and we test our data using a goodness of fit test. So we can run an exact test if we have two outcomes. And the exact test that we learned was a binomial test. Goodness of fit tests that we can run are the chi-square test or the g-tests. Both of them are okay. Different people have different opinions. And if all of our assumptions are met, if we have large expected counts and fairly equal counts, they don't have to be equal, but large sample sizes and nothing too drastically different in our sample sizes, then both chi-square and g-tests will work okay. With the g-test, I do note that you have to watch out for the zeros because of how I wrote that script. I wrote the script so that the observed categories is the one that's in the denominator. So just to show you, here's our script. I calculated our g by using the expected divided by the observed. So if we actually expected some items in a category, but we observed 0, this is going to fail. So you have to watch out for that. If we don't want to do classical tests, we can do randomized tests. And it's perfectly okay. And in fact, it's probably more powerful than both chi-square and g-tests alone because we generate our own null distribution using the underlying data. So here's our good summary. So another aspect of goodness of fit tests is to fit distributions, fitting discrete distributions. We learn discrete probabilities using the binomial distribution and the Poisson distribution. Now, when we introduced the Poisson distribution and binomial distribution, we talked about actually calculating observed probabilities and then generating expected counts. With the Poisson, we utilize it because Poisson's used for so many things in biology, including modeling spatial distribution of individuals across the landscape. With that Poisson distribution, we calculated a variance to mean ratio, or a coefficient of dispersion. If that coefficient of dispersion is equal to 1, then that means we have a random dispersion. It follows a Poisson distribution. If it's not equal to 1, then it does not follow a Poisson distribution. When we introduce these probabilities, and when I introduce this coefficient of dispersion, we never really explained how we can test if our coefficient of dispersion is actually equal to 1 or not equal to 1. Is it due to random chance that if we repeated our experiment we could get a different value, or is there actually something there that is making it not follow Poisson? So how would we do that? Well, we can do that by fitting our data to a Poisson distribution using a goodness of fit test. So this goodness of fit test is available in the VCD package. You may have to install this package if you are using a computer not in the computer lab. So I had to install this on my own laptop, and I normally have to install it anytime I update my computer. So with this package is called Visualizing Categorical Data Package, and there's a lot of things that we can use in this package. We'll use it for contingency analysis as well because I think that some of the displays are much better than the built-in package. What this package has is a function called goodFit. This is exactly what it does. It's a goodness of fit test where it'll take our table or vector of counts and fit it to a distribution or type of distribution. So the distributions that we can use would be Poisson binomial or negative binomial, and I believe there's other distributions that we can also use that's built in, but these are the common ones that we use. It also has this par part section, which is a parameters. So here we list the parameters that are required to be passed to the type. So with our Poisson, we have to pass it the lambda parameter. We have to tell it what lambda to use to generate our expected counts. Now, one thing we have to watch out for is the degrees of freedom, because now it comes down to this extrinsic versus intrinsic hypothesis. So everything up to this point has been extrinsic in nature. We've had this external idea of what our expected count should be. Well, we can have that here, and we can utilize the extrinsic hypothesis, which means the output that's given to us from the good fit function is usable. The degrees of freedom are correct. When would we have this? Well, when we generate our expected external to the data. So questions that extrinsic hypotheses would answer would be, does our data match what we expect? Is our statistic that we use equal to a given parameter? So is lambda equal to some value? Or is our probability of success equal to some value, and so forth? That's all things that are extrinsic in nature, and that means our good fit function will generate the correct p-values. But what happens if we just ask the question, is our data random? Do our trees follow a Poisson distribution? Meaning, are they randomly distributed across the landscape? Well, we didn't have a lambda, which means we're going to have to calculate lambda from the data itself. That's our intrinsic hypothesis part. We had to calculate lambda from the data in order to generate our expected counts. When we do that, we can't use the output from this good fit function because R doesn't know that we generated lambda from the data. So R is not going to adjust the degrees of freedom. So how do we adjust it? Well, the adjustment is that we lose 1 degree of freedom for everything that we estimate from our data. Our default is the sample size minus 1, so n minus 1, or the number of categories minus 1. And that's because we're going to fix our total count. So in our fruit fly data, where we had the white-eyed and the red-eyed flies, our expected was we calculated the sum of all of the flies that we counted. That was our estimate from the data, and then we used that to generate our expected. So that's our 1. And that's standard. That's standard for basically every test. Our degrees of freedom is our categories minus 1. If we're doing a goodness of fit test where we estimate lambda from our data, well, we lose 1 from the sum, but then we also lose 1 for estimating our lambda. So our k, our degrees of freedom, will be our number of groups minus 2. If we're fitting a binomial distribution, here if we estimate our probability of success, we lose a degree of freedom. So we lose 1 from our sum, from our total, and we lose 1 for that p that we estimate, so our degrees of freedom will be k minus 2. If we wanted to fit a normal distribution, we would have to estimate mu and sigma from our data. So those are 2 things that we estimate. So we lose 1 for the sum, we lose 1 for the mu, and we lose 1 for sigma, so our total degrees of freedom would be k minus 3. These would be the new degrees of freedom. And then what we'd have to do to generate our p-value is to do it manually. So we would have to use p chi-square, which is the area to the right of our chi-square value, and we would specify our degrees of freedom. And these would be the correct degrees of freedom. Alright, so before we get to the next slides, let's work through a random practice problem. So what we're going to do is generate some data. First, let's load the good VCD package. So I had to install the package, and I just did it here. I went to install, and then as I started typing VCD, it appears, and installed it, and it worked for me on Linux. Once it installed, I loaded the package, and it loads required package grid, so that works. So now, let's generate some data. So let's imagine we counted trees across a landscape. And we did this by dropping grids or setting up a grid and then counting the number of trees in each of the grids. So what we're going to do is generate random data. So we're going to generate it from a Poisson distribution, our poise. We're going to have 250 grids. We're going to say lambda is equal to 1.25. And I'm going to save that as dat. So then when I look at table dat, I run this chunk, and I get table dat. I get this. And your numbers are going to change because we didn't set the seed. But what I'm going to do is just pick one. That looks okay. And we're going to go with it. So you're going to have something similar, but again, your numbers are going to differ. So let's go through this extrinsic versus intrinsic hypothesis. So extrinsic would be answering a question like this. Is the average number of trees per grid equal to 1.5? Why is it extrinsic? Because we give you this 1.5 average. So what we're going to do is run the good fit function, and I'm going to save it as GF. So good fit, we're going to run the good fit on our table dat. Our type that we're going to run is Poisson, and then the parameter is going to be a list, and the list is lambda. That's what we have to give it. So this, if par is parameter, parameters are always presented as a list, and we separate items based on a comma. So we're going to fit a Poisson where lambda is equal to 1.5. Now, if I just run this good fit function, I'm going to highlight that and run it. Oops, sorry. I'm going to highlight and just run this. You will see that this gives us both the observed and fitted functions. So this is my observed, this is our expected counts. We can do the same thing by using the dpoise function. So dpoise, mine is 0 to 5. Our lambda is 1.5. That's our expected counts. We have 250 items in there, so these represent our expected. So good fit does all of this for us. So we don't need this. Alright, so that's our good fit. Now this doesn't run the chi-square test yet. So what I'm going to do is save this goodness of fit table, good fit table, and then what we are going to do is view a summary of it. So saves the goodness of fit table, and then it reports the summary. So here, this is my output that I get. So it gives me two lines. It gives me Pearson and it gives me a likelihood ratio. This Pearson line, that is our Pearson's chi-square test. So if I ran a chi-square, I'd report it as 17.3, degrees of freedom of 5, p-value 0.003955. If I wanted to run a g-test, that's this line. That's our likelihood ratio. So there's nothing different. We don't have to do anything different to run a g-test. It is given to us. It is given to us here. So what's our write-up? Well, let's look at our p-value. Is it greater than 0.05 or less than? It's less than, so we reject our hypothesis. So our observed does not match our expected. Our expected were derived assuming our lambda is equal to 1.5. So is it equal to 1.5? No, it's not. The average number of trees per grid is not equal to 1.5. And I can report the chi-square test, 17.3. Degrees of freedom is 5, and p-value is less than 0.004. That's our chi-square test. If I wanted to report the g-test, I can. My g is equal to 20.88. I will say 20.9. Degrees of freedom is 5. This one 0.0009. So both ways. If we do a chi-square or a g-test for fitting distributions, it's run the same way. We just read a different line. So that's extrinsic. And we know it's right because when we look at how many categories we saw 5. So 1, 2, 3, 4, 5, 6 when we include the 0. So our number of categories will be 6 minus 1 or 5. But what if we ask the question, are the trees randomly distributed across the landscape? Well, now we didn't specify a lambda, so we have to calculate it. So if we do mean of our data vector, we get a 1.18 lambda. So what we would do is run our same goodness of fit test. So GF is good fit. We're going to use our data vector, table dat. Our type is Poisson. The parameters we have to give it, and our lambda that we're going to use will be the mean of our data. And then when we view the summary, we'll look at our information. So here it gives us the same degrees of freedom. No big surprise. We have the same number. We've got 6 categories. Minus 1 equals degrees of freedom of 5. But that is not correct. What we have to do is get the correct p-value. And we do that by using p chi-square. We give it our chi-square value. So 1.847977. Our degrees of freedom is 4. So we lose 1 degree of freedom because we estimated our lambda from the data. And our lower dot tail equals false because we want our upper end. So our p-value isn't 0.8697. It's actually this value. So is the data random? Yep. Data appear to be randomly distributed. Here I did chi-square test. Chi-square is equal to 1.85. Degrees of freedom is 4. And our p-value, 0.76. The g-test does the same thing. If we wanted to run the g-test, so this will be the correct p-value for the g-test. G-test follows our chi-square test statistic with the appropriate degrees of freedom. So it would be 2.455039, degrees of freedom 4, lower dot tail equals false. I run it. I get that p-value. Our write-up's going to be the same thing, with the exception of replacing chi-square with our g-tests and our numbers. So 2.46, degrees of freedom 4, p-value is 0.65. So with the intrinsic, we can use the table that's generated for us. So here's our extrinsic hypothesis. We use the table that's as reported. If we're doing intrinsic hypothesis, we're going to have to look at the degrees of freedom, look at how many groups we have, and correct our p-value using the correct degrees of freedom. I also want to note now that in these problems, I get this notice. Chi-square approximation may be incorrect. The reason it does this is because we're getting R will automatically detect whenever we have a small sample size in the expecteds. When we have expected cells that are less than 1 and they are being used to calculate the chi-square test statistic, then R will give us a warning. This is usually our sign that we're violating a chi-square test, and we should prefer a g-test. As we continue doing these, though, as you'll see, perhaps we can't just do a g-test. Perhaps we have to combine groups to fix this issue. That's beyond the scope of this course, but when you see this, you should be looking back at your assumptions. All right, so here's an example problem. We've got number of trees per grid. They were recorded in an ecology course. Here's our frequency table. All right, so our question 1 is, do the data follow a Poisson distribution with an average of two trees per grid? And question 2, are the data randomly distributed and follow a Poisson distribution? So what I want you to do is run the tests and answer these 2 questions. So pause the video, go back and do this. Use what we've done, what we just did as a guide, and answer these 2 questions. All right, so hopefully you had a fairly easy time. Red box asks the question, how are these 2 questions different? Well, it comes down to intrinsic versus extrinsic hypotheses. So this is extrinsic. I give you the lambda, 2 trees per grid. This is intrinsic. You have to calculate lambda from the data. So for question 2, you will have to adjust your p-value. So for question 1, do the data follow a Poisson distribution with an average of 2 trees per grid? So lambda is given in the problem, so we don't have to calculate it. But what we do have to do before we can run the good fit is to convert these data, these frequency data, into a vector. So you can do that using the rep function. So if you didn't do that, if you were stumped, go back and try. Now pause the video and try to do that. Use the rep function to create a vector from this frequency table. This will be a very good review for the final exam. All right, so hopefully you have your vector, and then once you have the vector, you can run the good fit function and view the summary. So here's how I did it. So here's how I used the rep function. I did trees and then grids, and then I called it data1. So rep trees comma grids, data1. I passed data1 to the good fit. Notice I didn't have to use the table. I do by convention, but I wanted to show here that you do not have to use the table. R will generate the table by itself. We're fitting a Poisson distribution. Parameters that we have to pass is lambda. And this is our G fit. This is our summary table. So there's our observed. These are our expected. And these are the Pearson residuals. So these would be the chi-square values that we would calculate if we did this in Excel. So then once we have this, once we have G fit, all we have to do is look at the summary of that G fit to get these numbers. So again, here's our chi-square. That's our Pearson line. And the second line, likelihood ratio, that's the results of our g-test. So we know our lambda, we gave it, so degrees of freedom will be correct. Note, we also get a warning message here, and that warning message is likely due to this category. So we saw less than 1, or we expect less than 1. That violates our chi-square. That's why it produces a warning message. So for this problem, we should probably use the g-test. So is lambda equal to 2? Yep, for these data, it looks like lambda is equal to 2. So trees appear to follow Poisson distribution with a mean of 2. I use my g-test. I give the g-test statistic, degrees of freedom 7, and the p-value of 0.81. Question 2 is basically the same thing, but this time I use the mean of our data vector for lambda. So generate my good fit, and then you can look at the good fit, and again, These are the chi-squares. All right, here's our expected, assuming that our lambda is equal to the mean of our data set. All right, this is less than 1, so I'm going to expect to see a warning message. And then once we have the G fit, I'm going to look at the summary, and I get this output. So again, Pearson, that's our chi-square line. Likelihood ratio, that's our G-test line. But our degrees of freedom are now incorrect. These represent the number of categories that we have, so 8 minus 1. But we have to subtract 1 because we estimated lambda. So what we're going to do is take our G-test, our G right here, 0.6838, and pass that to the p chi-square with the correct degrees of freedom. So df equals 6 and lower dot tail equals f, and that gives us the correct p-value to report. So again, are the trees randomly distributed across the landscape? Yep, they are. They are. P-value is greater than 0.05, so our observed matches what we expect. So hopefully, with the examples that we've been going over, you feel comfortable doing these problems. So here is one practice problem that you can do on your own. So this was from actual data from an ecology class in lab. And what we did was measured ragweed in a small area of grass. So we dropped grids, we counted the number of ragweed in each individual grid, and this is our frequency table. So our question, are ragweed randomly dispersed or distributed in this grass area? So perform the correct test, answer the question, and then write it. Have a sentence that writes up your result. So pause the video, do that. And pay attention. Getting these data in, and then also, are the degrees of freedom correct? All right. So practice problem 1. We're asking, are these ragweed randomly distributed across the landscape? So the first thing we have to do is convert our frequency table into a vector so that we can analyze it in R. So here's how I convert a frequency table to a vector. Create my X frequency, I called it ragweed. Ragweed's available. And now I want to run the goodness of fit test. So the goodness of fit test, I'm going to use good fit. We're fitting it to a Poisson. And I didn't specify a lambda. So we're going to have to calculate it from the data. So I run good fit function. I saved it as GF, and now I'm going to look at my GF. And what I can see is that I have 11 categories, so 0 all the way up to 10. Our degrees of freedom is going to be 9, but we have to subtract 1 because we estimated lambda. So when we run the summary, that 10 degrees of freedom is incorrect. Our correct degrees of freedom should be 9. So what we're going to have to do is obtain the correct p-value. Now, which one should we use? Should we use Pearson's chi-square or G? Well, notice these are two drastically different values. And the reason they're drastically different is related to our expected counts. So these values here are all less than 1. Anytime we have less than 1, we violate our chi-square. So because of that, I'm going to favor our G-test. So I'm going to use the G-test statistic. Degrees of freedom that we are working with is 9. So 10 minus 1 is 9. And we get a p-value of 1.62 times 10 to the negative 15. So is our ragweed randomly distributed? No. They're not randomly distributed because they did not follow a Poisson distribution. Our observed did not match what we expected from a Poisson distribution. We ran a g-test. Our g is 90.00. Degrees of freedom is 9, and our p-value is much less than 0.0001. Now, if it does not follow a random distribution, then what does it follow? Well, now we can go back to that coefficient of dispersion. It's our variance to mean ratio. So we know it's not randomly distributed, so the variance to mean ratio is not equal to 1, and thus we can say the ragweeds are not randomly distributed. They are aggregated or clumped. And we know that because of the variance to mean ratio. So we just kind of took it that next step. So here's practice problem number 2. You recorded the number of males in turtle nests. In this population of turtles, 7 eggs always hatch, and sex appears to be determined by nest temperatures. So these tests may have been exposed to full sun. Thus, we're going to ask, is the probability of being male equal to 35%? So we're not fitting it to a Poisson anymore. We're going to fit it to a different distribution. So our distribution we're going to use the good fit function, but we're going to fit it to a different distribution. So pause the video, see if you can work this out using the good fit function. If it helps, you can go back to the discrete distribution, discrete probability presentations, because we had some examples like this. So first thing we have to do with this practice problem is convert the data into a vector. Did that here. And then we run the goodness of fit test. So with our good fit test, we are fitting a binomial distribution. It's either male or female. And with the binomial distribution, we have to give it the out of number, so size is equal to 7, and we have to give it the probability of success, so prob equals 0.35. When we run this, it does the goodness of fit test and then generates our observed and expected tables. So what we then do is take that table and pass it to our chi-square. Now, our chi-square approximation may be incorrect because, again, when we look at our expected counts, we have this category here where we saw 0, and we actually fitted a value less than 1, or we expected something less than 1. So we get our warning that says it may be incorrect, and that should be concerning. Also, when we see degrees of freedom that differ, we should know something's up with our table. So I'm going to run the entire chunk so that they're up together. So with the likelihood ratio test, the g-test, what it's going to do is to look at a 0. If it has a 0, it's going to usually exclude it because that observed is likely in the denominator. So that good g-test basically takes that value and drop it completely out. Now we can still do the chi-square test, but note that we may be incorrect, or we can still use the g-test. If we do the g-test, we will have to go back and manually enter these terms, or we can deal with it in our write-up. And in this case, I'm just going to deal with it in our write-up. So a g-test was performed to determine if the probability of being male is equal to 35. Because 0 clutches consisted of 7 males, the test excluded 7 male category. So that kind of deals with our reduction of degrees of freedom. So the probability of having a male offspring, is it equal or not equal? Well, it's based on our p-value. So I would say it's not equal to 0.35. Our g is 13.20. Degrees of freedom is 6. And our p-value is less than 0.04. So that brings us to small observed and small expected values. So the good fit will take a table or take a data set and generate our table. And it's possible that we have a rare event that occurs, or that we have something that should be not super rare, just doesn't occur. So we end up with a very small O's or very small expected values. When this happens, g-tests are usually a little bit better because they handle those small values, and our p-values tend to be a little bit more realistic. But if we wanted to be a little bit more specific, or if we're dealing with data sets, data tables that have a lot of these small values, then what we have to do is take corrective measures. And the corrective measures that we're going to have to do is basically combine our groups so that our expected counts exceed 1 for all of our groups. And we also have to make sure that 20% of our groups exceed that 20% too. So once we do this, then we would have to manually rerun the analysis using our grouped categories. So as an example of combining categories, I have this code that I want you to run. So we're going to set the seed, run the R negative binomial, all right, in our table. So table, you'll look at our table. So what this will do is we're going to assume that the data are ragweed per grid. All right, something that we've been doing through this class numerous times, and we're going to ask: are these plants randomly distributed? So what we want to do is fit these data to a Poisson distribution, and we're going to calculate lambda from our data and everything else. So what I want you to do is, with these table, I want you to write down the chi-square observed and the g observed value. All right, and then look at the p-value and see, does our conclusions differ? And then, after you do that, I want you to look at the expected numbers. So pause the video and go ahead and do this in R. Alright, so when I did this, this is my goodness of fit table. So I did the same thing. gfit, did Poisson, type equals Poisson, par equals the list, and then lambda equals mean of my vector, so mean of our cat. All right, and this is my gfit table. So what I noticed is not only did I get a warning that chi-square approximations may be incorrect, but when I look at the fitted table, I see that there are several categories, basically 9 and beyond, that have very small probabilities, yet I did observe some of these data points. So what I'm going to have to do is take a corrective measure and basically combine these groups into a 9 or more category. And it's going to be 9 or more as long as our expecteds are greater than 1. Now when I do that, you'll see that the sum isn't quite 1, so perhaps what I need to do is add one more category, one more to 8. So that's what I'm going to do. So I can access those by taking the sum of the gf.fit, so that's our goodness of fit table. $fitted gives this vector only, and then subscript notation says give me rows 9 to 17. Well, we're going to use 8 to 17. So we're going to take our fitted values. We're going to have all of our fitted values from 1 to 7, and then we're going to add up all of the fitted values from 8 to 17, so all of the rest. All right, so these are the same values, 21, 47, those are the same values here, and then I get the 8 or more category, and we saw 1.6555 in that 8 or more. I have to do the same thing with our table. So our observed values are going to be all of these up until I get the 8, and then I'm going to collapse these values. So 6 plus 6 plus 3 plus 2 plus 2. And then I'm going to manually run my chi-square test. So the chi-square test, I'm going to use our observed values, where the p is our fitted value vector divided by the sum of our fitted values. So it's going to take the sum of this, take this vector and divide it by the sum, each of these, divided by the sum to convert to a probability, and we're going to do correct equals false. All right, and we get the chi-square output, and it says, yeah, it could be small. All right, but notice the degrees of freedom. So it gives us 7, which we have 7 categories, but remember, we calculated lambda from our data set, so we're going to have to go back and correct it. So use the p chi-squared to do that. This is how we handle small samples. Are we going to do this in this class? No. I'm not going to ask you to do it on the homework. I'm not going to ask you to do it on the final. However, this is getting to be the proper way to do it. In the grad stats class and future statistics classes, this becomes very important to do. We just won't do it. I present this for completeness. So that wraps up the one categorical variable. What's next is to look at situations when we have two categorical variables.